Hello and welcome to Nikolai's genetics lessons, and today's problem is the molecular weight of unspecified protein and the physiological conditions is 70,000 Dalton as determined by sedimentation equilibrium measurements and by the gel filtration chromatography. The SDS polyacrylamide-gel electrophoresis of the protein yields a single band corresponding to the molecular weight of 70,000 Dalton. However, in the presence of the reducing agent beta-mercaptoethanol, the Sds-Page shows two bands corresponding to the molecular weights of 30,000 and 20,000 Dalton. From this data, describe the native protein in terms of the number of subunits present, the molecular weight, and the kinds of bonding, covalent or non-covalent, existing between these subunits. This problem on the first glance looks like it is a mystery, because we have one band which represents 70,000 Dalton protein, then we use some chemical which supposedly have to break it into smaller subunits, and as a result we have one band that corresponds with the protein which weighs 30,000 Daltons and another band which corresponds to the protein which weighs 20,000 Daltons, and together 30 plus 20 would make 50,000, and looks like we lost like 30% of the original mass of the protein. Actually, this is very simple problem. Take a look. We're using reducing agent, which is beta-mercaptoethanol, which breaks disulfide bridges between polypeptide chains. Let's say that one protein consists of 3 polypeptide chains, and let's say that here between these 3 polypeptide chains we have disulfide bridges like this. So one disulfide bridge, and here's another one, and we may have another one here. It doesn't matter how many disulfide bridges hold these 3 polypeptide chains, and we perceive this as single protein. When we use reducing agent beta-mercaptoethanol, all these bridges would be broken. These disulfide bridges are formed by R groups of 2 cysteines, and cysteine R group is going to be CH2SH, and here is going to be another one CH2 and SH. I show these R groups now in reduced form, and when R groups are going to be oxidized, we are going to see the following picture: CH2 sulfur, sulfur, and H2C here. So now we see that 2 cysteines formed a disulfide bond, but this oxidation reaction is reversible with reducing agent beta-mercaptoethanol. This disulfide bridge, this covalent bond, is going to be broken, and again all these 3 polypeptides will separate. And what we are going to see, imagine that we now run a gel and we load our sample here in the well, and when we apply electrical current, these 3 polypeptide chains would move according to their size, according to their molecular weight. And if you expect to see here 3 bands, it's not usually the case. Imagine that this top polypeptide strand is 20,000 Daltons, and this strand in the middle is 30,000 Daltons, and strand on the bottom is 20,000 Daltons. In this case, we will see that this strand would make one band, let's say somewhere here, and another strand, which is here, would move faster, it is smaller, and let's say would make a band here. And this strand, because molecular weight is the same as this one, also would move with the same speed and also would end up here, and instead of 3 bands we are going to see only 2 bands here, but in this band 2 bands are hidden. As you see, this picture perfectly explains why we see only 2 bands. One would be 30,000 Dalton polypeptide chain here in the middle, and this 2 would make another band which is going to be 20,000 Daltons, but is going to make only 1 band. And this is what we see in our problem. One band 30,000 and another is 20,000, but actually in this band there are 2 polypeptide chains, and if, for example, this polypeptide chain would be any other weight, then we would see 3 bands instead of 2. So now you see there is no any mathematical mystery here, and this is all for today. Thank you for your attention and see you in the next video. Goodbye.